presentation I gave in the lab.
Yeah.
I think it was more simplified now, the
Yeah.
version.
Okay, that's super. Okay, right. Recording started. Great. Okay.
See what I can share here. Screen. Then. So can you see it?
Yeah.
Okay. So I'm going to speak a little bit about the data that I'm working with. It's a data that was recorded by Anderson, was a PhD of Mark Cunningham. So this data was recorded from human tissue. The humans in this case, they were humans that were undergone under surgery because they had a glioma of some sort of glioma, which was a glioma uh, that made them to have epilepsy. So they couldn't be treated by medicine, what's called refractory epilepsy. So they need to undergo the surgery to remove the glioma and treat both the glioma and epilepsy. So the main aims of this introductory presentation is to show an introduction of spark sorting, the methodology that I use, the LFP data, the spike data, and what we want to do next. So to record spikes in a tissue, so if you imagine this figure in the left, it would be the tissue, and the tetrode would be 4 electrodes that go inside the tissue, and the neurons are around these electrodes. So you have a small region, if you see in the second figure here, the blue region, where the electrodes can record perfect waveforms. So these waveforms, we can cluster them, and each waveform, each neuron has a specific number of channels and type of channels. So the waveform of these neurons would be unique for these neurons. So we can separate unique neurons based on the waveform. And if you go a little bit further, you still can get some waveforms, but they will be quite small and it would be hard to define them. So we call the first region around the electrode, the region that you can collect single unit activities. And the big region, like the blue region, we can collect multi-unit active. They are both important, and depends on the experiment. You want to look for the population of neurons, so you look for multi-unit active, or you want to look for single neurons, then you go directly and separate these single neurons. And we can filter this data in low frequency and record the low frequency local field potential, which can show how the entire network varies in time during this recording. So I'm going to show how we cluster these neurons and select the single neurons. So single neurons, they have a very high frequency. So we need to filter up this data. So pass a high pass filter or a band pass filter around 300 to 7 kilohertz. The noise has to be removed. And then once the noise is removed, you can threshold it. And what you finish with is some waveforms. And then you do PCA, which is a type of forget now, and cluster. So PCA would transform this data in another base. So usually it spread the data. And so once you have this data spread, you can cluster it using some cluster technique. So usually we did this manually. You run one script for each, and now some people create scripts that can do it automatically. We still have to go and look for these clusters and these spikes to see if they're just noise or if they're real spikes. And the data that we record from these humans, they come from these probes. So these are multi probes. So they have 8 chunks, and in each chunks they have 8 electrodes. These 8 electrodes. So in the total, we have 64 channels. And these channels would record neurons around them. So the idea is when you have more than one channel, you can discriminate better the waveform of the neurons. So that's why we cluster more channels in one electrode. And the data was processed and run to the clustering software, and we finish with spikes and waveforms. So this is the visualization of the software after you cluster it. So you have these waveforms in blue, the small one, and we have to check manually and classify them in spikes or noise or multi-unit active. So we have different ways to do this. One way, but most of them by eye. And that's why we plot different properties of this neuron to check if it's a neuron or if it's just a noise. So in this case, is a well isolated neuron, and you can see that the waveform is perfect, and there are other properties here. So I'm going to tell just about the property here. So the neuron, once it spikes, it can't spike again for the next millisecond or two. So we can plot the autocorrelation and check the time histogram of the spikes. And if you see spikes between 1 to 2 milliseconds, it would mean that it's another neuron and the sorting is contaminated. So then we don't consider it as a clean neuron. Here. Here is an example of a noise. So although the clustering software collected, it's just noise that passed the threshold. So as you can see, the waveform doesn't look like a single neuron. And most of the waveforms was cut by the noise threshold. And there are 61 neurons spikes around the 2 milliseconds. So it's a noise neuron. And also we can compare them and see if they're the same neurons split in different clusters. And if they're the same, we have to put them together. And also, once you get the data, you can filter it using a low pass filter. And when you filter the data in a low pass filter, what you finish is with a type of low field potential, local field potential, which demonstrates how the network varies, would be similar to EEG recording in humans. And so we can see the seizure here because the seizures, so these are examples of epileptic seizures, they have very low frequency. And what we want to look after, it's how the neurons, the single neurons here in the left, how they spike in relation to the seizures. So we have the spike times and the seizures. We want to look how they spike around the seizures. So if you do a zoom here, you can see that around the seizure, which is this conglomerate of black lines, you have a very interesting pattern of spikes. And when we go, so we want to do first is look at this Elahian et al. 2018. So what I'm trying to do now is to replicate this paper. So part of it I have done, which is the clustering, and I have also separate this neuron using the same method of he's using. So what he's doing here, he got the seizures and separated single units into inhibitory and excitatory based on waveform. He clustered it, and then what he's looking at is how these neurons spike around the start of the seizure, the beginning of the seizure. So what I think we could do with these students is get some part of this data, and we try to look with probably not t-test, but z-test, because it's too many spikes, but it's very similar to t-tests, and look for the spikes before and after the seizure onset, or the difference between the inhibitors, excitators, and we can check if there's some difference here. So what I have marked the data, I marked them on before the start of the seizure, on the start of the seizure, and the end of the seizure. So I have the times of the inhibitory and excitatory neurons. So then I think you can pass this to the students, and the students look at how these neurons spike before, 10 seconds before, 10 seconds after, or during the seizure and before the seizure. So I think that's what I thought for them. And I think that's all. And
yeah,
thank you.
that sounds good. Yeah. So, I mean, I'll just make a couple of comments, just to sort of, first of all, thanks, Matthias. That's brilliant, and hopefully the students can appreciate that these are incredibly rare data sets. So just to kind of give them a bit more background. So what we do in the lab is we are able to study how neurons interact with one another, and we do that using a particular type of preparation, which is called a brain slice. So students are probably thinking, what do they mean by recordings in human brain tissue? So as Matthias sort of mentioned, these are patients that have a brain tumor, and in many cases the brain tumor will cause epilepsy to occur. Now, it's not always possible to stop the tumor from causing seizures by using conventional therapies like chemotherapy or radiotherapy. And actually what is the preferred method of treatment is, if you can, to remove the tumor, to go in and do an operation, a craniotomy, that allows the neurosurgeon to remove the tumor. Now, in removing the tumor, what the surgeon normally has to do is also remove a little bit of brain tissue around the tumor, which is called the peritumoral region. And that peritumoral region will contain the tumor cells, but it will also contain some kind of, I don't want to call them normal because the surgeons don't want to take out normal tissue, but it will contain some kind of normal brain cells as well. It's a sort of margin that the surgeons have to remove. And it's in that peritumoral area, this kind of ring of tissue around the tumor, that the seizures actually originate from. So that's where the seizures are being generated from. The tumor mass itself does not generate seizures because it's just tumor cells. And they are not, at least we don't think they're electrically active. But remember, it's the brain cells that are electrically active. And that then leads me on to just a kind of brief sort of, I suppose, clarification. So when we're talking about, or when Matthias is talking about spiking, what that means is, what he means there is, that is a neuron, a brain cell, generating an action potential. So you're hopefully all familiar with how neurons behave, and the currency of information that neurons, brain cells, work upon is this idea that they can either fire an action potential or they don't fire an action potential. So remember, an action potential is an all or nothing event. It either happens or it doesn't. And what we know is that during an excessive period of activity, such as a seizure, so this is one way to think of it, is it's kind of like an electrical storm that takes place in the brain. You have different types of neurons contributing to that seizure activity. Now, a further statement to make on that is that in the brain, one way to think about the brain is that it's a little bit like a seesaw. And on one side, you have excitation, and on the other side, you have inhibition. And in our brains right now, that balance of excitation and inhibition is okay. But in somebody that has a seizure or is suffering from epilepsy, that EI balance becomes essentially, it becomes unbalanced. And you can either have too much excitation or not enough inhibition. Now, what we know is that brain cells, neurons, can be classified both in terms of their physiology and their chemistry, well, their neurochemistry, into cells that are excitatory. These are sometimes referred to as pyramidal neurons, or inhibitory interneurons, which are the inhibitory cells. And the excitatory pyramidal cells use glutamate as a neurotransmitter, and the inhibitory cells use GABA as their neurotransmitter, GABA. So that's just a few sort of additional comments just to make in terms of trying to understand a little bit more of the neurobiology that's taken place within the data sets that Matthias is taking you through. Does anybody have any questions?
Mark, just one thing, the neuro part of the neurons and the nervous system part of the A-level biology, yeah, is A2, is next year, but I will direct them even to the SIA website where they can read a little bit, yeah, about the neural system there on the A-level syllabus. I'll send them that information. Okay. I mean, I think probably with this project, it's going to rely a lot on on data analysis, and obviously the data is ready. It's been cleaned by Matthias. It's been prepared, but it's probably just worth helping them kind of contextualize, what it actually means in terms of the signals. In terms of when we talk about a spike, that actually means the action potential signal that's being generated by a single neuron that was very close to that array that we placed in the tissue. And this is the live tissue. I suppose that's another important thing to get across to the students, that when we get this tissue from the surgeons, so we collect the tissue from the surgeons, it's been consented, so the patients are happy for us to use the human tissue for scientific experiments. That's another important thing to state. This is patient material, and of course it has to be consented. We have to make sure that patients are consented. We don't know anything about the patients because that's all been anonymized. But we collect the tissue, and then the tissue is put in a physiological solution. It's kind of like the way I describe it is it's kind of like a brain juice, and it's got all the goodies in there that keeps the tissue alive. So it actually allows the neurons in the tissue to keep firing, allowing them to communicate with one another through the neurotransmitter systems that I've already described. And what we do is we have these specialized chambers in the lab that allow us to continually perfuse these very thin slices of brain tissue. So they're 450 microns thick. So that's just under 0.5 of a millimeter. So we can get this brain juice into those thin sections of tissue, human tissue, and we can also
keep it at physiological temperatures, so around about sort of 33 degrees centigrade, and we can make sure that the tissue is oxygenated as well. So we fooled that little sliver of human brain tissue into thinking that it's kind of never left the whole brain, by sort of recreating the physiological conditions that allow it to still operate and to generate those signals. And those signals are manifest in terms of, its ability to generate seizure activity. So in a bath ex vivo, as we sometimes refer to, and also by the fact that we can record the spiking activity that underlies the generation of that seizure activity as well. So hopefully that gives a bit of an overview in terms of the biology of the data that you're going to be using, where it's come from, and also what you're going to be doing in terms of, as Matthias has laid out, looking at the behavior of these discrete populations of neurons before and after the seizure has started. And I think, that's quite a nice project. Matthias, you mentioned the Z test as an additional statistical method that we would want to use. Kieran, are you familiar with that one?
No, Mark.
Okay,
but,
so
that won't be a problem.
Yeah. I mean, Matthias, if you've got some information on that, maybe you can pass that on to Kieran as well. I think you're muted, Kieran, or you're muted, Matthias, sorry.
It's very similar with t-test, but t-test is not recommended when you have more than 30, 35 samples.
Right, yeah.
So you have to use another table for the 5%.
Okay.
Instead of the T table, use the Z table.
Right. Okay.
So it's the same test, Matthias, only you're using a different table.
Yeah, exactly. It's very similar. You test populations, different between populations.
Yeah.
Okay.
Because these are huge. We could have hundreds of data points. So yeah, and then we'll send you some information on the Z test.
Yep.
We'll send some papers, some references with respect to the literature review. Matthias and I can do that. And then the other thing, Matthias, I just wanted to ask is making sure that the students will have access to the data. So the data at the minute is, where have you got that? Have you got it on the cloud somewhere?
I can share perhaps in Google Drive.
Yes.
If
would that work, Kieran?
I have a lot of the students that are using the Google Drive in different projects, so I don't think that would be a problem at all.
Okay, that's perfect.
Good. And then Matthias, are you happy to maybe organize a tutorial then with the students in terms of maybe sitting down and just kind of orientating them with the data and what you exactly want them to do with respect to the data?
Yeah, no problem at all.
I can sit in on that as well. So you and I can sort of look at our diaries and then just liaise with the students in terms of setting a date for this. So Matthias, these projects run over, it's the next 6 weeks, Kieran, is that right?
No, Mark, 4 weeks.
4 weeks.
4 weeks from yesterday.
4 weeks from yesterday. Okay. So again, we don't want to overload them too much. So maybe what we can do is, because you're going to cover the chi-squared, and maybe we can do the Z test with them this week as well, Kieran.
I will, Mark, yeah.
And we'll send them the references, and then if maybe we if we meet them next week to do this kind of initial tutorial in terms of the orientating them with the data sets and what we want them to do. Does that sound reasonable? So maybe for next Monday?